The overall goal of this method is to purify endodermal cells generated from human embryonic stem, or ES cells.
This method can help answer key questions in the stem cell field about endodermal differentiation.
The main advantage of this technique is that a pure endoderm cell population can be obtained after the removal of undifferentiated cells.
Before beginning the procedure, coat 6 well cell culture plates with 1 milliliter of basement membrane matrix per well for at least 30 minutes at room temperature. Then, to harvest the human ES cells, aspirate the medium from an 80 to 90% confluent human embryonic stem cell culture with a sterile glass Pasteur pipette, and wash the cells with 2 milliliters of PBS per well, shaking the plate, and aspirating the saline to remove the dead cells and debris. Next, gently dissociate the cell clusters with 1 milliliter of enzyme-free passaging solution reagent at 37 degrees Celsius and 5% carbon dioxide until the cells exhibit clear signs of disruption into smaller clusters. After about 7 minutes, add 1 milliliter of DMF12 medium to the wells and pipette up and down a few times with a 1 milliliter pipette tip to detach the remaining cell aggregates into a single cell suspension. Using the same pipette, transfer the cells into a centrifugation tube and rinse the wells with 1 milliliter of fresh DMF.